Hello, welcome to Biology, the scientific study of life. Again, more about proteins. These are some functions of membrane proteins. Let's begin with first one, transport. Notice the left. A protein that spans the membrane may provide a hydrophilic channel across the membrane that is selective for a particular solid. The right one is other transport proteins shuttle a substance from one side to the other by changing shape. Some of these proteins hydrolyze the ATP as an energy source or to actively pump substances across the membrane. The second one, notice that enzymatic activity. A protein built into the membrane may be an enzyme with its active site exposed to substances in the adjacent solution. In some cases, several enzymes in a membrane are organized as a team that carries out sequential steps of a metabolic pathway. Third one is signal. Notice the signal transduction. A membrane protein may have a binding site with a specific shape that fits the shape of a chemical messenger, such as hormone. The external messenger signal may cause a conformational change in the protein receptor that relays the message to the inside of the cell. Notice that. Fourth, also function cell recognition. Some glycoproteins serve as identification tags that are specifically recognized by other cells. Function number 5. Intercellular joining. Membrane proteins of adjacent cells may hook together in various kinds of junctions, such as gap junctions or tight junctions. Notice that picture. The last function, attachment to the cytoskeleton and extracellular matrix, ESM. Microfilaments or other elements of the cytoskeleton may be bonded to membrane proteins, a function that helps maintain cell shape and stabilizes the location of certain membrane proteins. Proteins that adhere to the ESM can coordinate extracellular and intercellular changes. More about the role of membrane carbohydrates in cell recognition. Cell recognition, a cell's ability to distinguish one type to neighboring cell from another, is crucial to the functioning of an organism. It's important, for example, in the sorting of cells into tissues and organs in an animal embryos. It's also the basis of the rejection of foreign cells, including those of transplanted organisms, by the immune system, an important line of defense in vertebrate animals. Notice this one. The way cells recognize other cells is by binding to surface molecules, often carbohydrates on the plasma membrane. Membrane carbohydrates are usually short branched chains of fewer than 15 sugar units. Some of these carbohydrates are covalently bonded to lipids, forming molecules called glycolipids. Recall that glyco refers to the presence of carbohydrate. Most, however, are covalently bonded to proteins, which are thereby glycoproteins. The carbohydrates on the external side of a plasma membrane vary from species to species, among individuals of the same species, and even from the one cell type to another in a single individual. The diversity of the molecules and their location on the cell's surface enable membrane carbohydrates to function as markers that distinguish one cell from another. For example, the 4 human blood types designed A, B, AB, and O reflect variation in the carbohydrates on the surface of red blood cells. Synthesis and thiness of membranes. Membranes have distinct inside and outside faces. The two lipid layers may differ in specific lipid composition, and each protein has directional orientation in the membrane. Notice that when a vesicle fuses with a plasma membrane, the outside layer of the vesicle becomes continuous with the cytoplasm layer of the plasma membrane. Therefore, molecules that start, notice number one, start out on the inside face of the ER end up on the outside face of the plasma membrane. The process shown here starts with one as the synthesis of membrane proteins and lipids in the endoplasmic reticulum. Carbohydrates, green represents green one, carbohydrates, notice that, are added to the proteins parable, notice parable, proteins, notice, making them glycoproteins. The carbohydrate proteins or the carbohydrate portions may then be modified. Inside the Golgi apparatus, the glycoproteins undergo further carbohydrate modification, and lipids acquire carbohydrates, becoming glycolipids. Transmembrane 3, notice stage 3 here. Transmembrane protein parable dumbbells, membrane glycolipids and secretory proteins, notice the parable spheres, are transported in vesicles to the plasma membrane. Therefore, notice the fourth stage number 4. There the vesicle fuse with the membrane, releasing secretory proteins from the cell. Vesicle fusion positions the carbohydrate of membrane. Notice also outside membrane glycoproteins and glycolipids on the outside of the plasma membrane. Thus, the asymmetrical distribution of proteins, lipids, and their associated carbohydrates in the plasma membrane is determined as the membrane is being built by the ER and Golgi apparatus. More about membrane structure results is selective permeability. Again, membrane structure results in selective permeability. The biological membrane is the ability to regulate transport across cellular boundaries, a functional essential to the cell's extents. We will see once again that from this function, the fluid mosaic model helps explain how membrane regulate the cell's molecular traffic. A steady traffic of small molecules and ions moves across the plasma membrane in both directions. Consider the chemical exchanges between the muscle cells and the extracellular fluid that bathes it. Sugars, amino acids, and other nutrients enter the cell, and metabolic waste products leave it. The cell takes in oxygen for cellular respiration and expels carbon dioxide. It also regulates its concentrations of inorganic ions such as Na+, K+, Ca2+, and Cl- by shuttling them one way or the other across the plasma membrane. Although traffic through the membrane is extensive, cell membranes are selectively permeable, and substances do not cross the barrier and discriminate. The cell is able to take up many varieties of small molecules and ions and exclude others. Moreover, substances that move through the membrane do so at different rates. The permeability of the lipid bilayer. Hydrophobic nonpolar molecules such as hydrocarbons, carbon dioxide, and oxygen can dissolve in the lipid bilayer of the membrane and cross it with ease without the aid of membrane proteins. However, the hydrophobic core of the membrane impedes the direct passage of ions and polar molecules, which are hydrophilic through the membrane. Polar molecules such as glucose and other sugars pass only slowly through the lipid bilayer, and even water, an extremely small polar molecule, does not cross very rapidly a charged atom or molecule and its surrounding shell of water. Notice that boom. Transport proteins. Cell membranes are permeable to specific ions and a variety of polar molecules. These hydrophilic substances can avoid contact with the lipid bilayer by passing through transport proteins that span the membrane. Some transport proteins, called channel proteins, function by having a hydrophilic channel that certain molecules or atomic ions use as tunnel through the membrane. For example, the passage of water molecules through the membrane in certain cells is greatly facilitated by channel proteins known as aquaporins. Other transport proteins, called carrier proteins, hold into their passengers and change shape in a way that shuttles them across the membrane. Notice also in both cases the transport protein is specific for the substance is translocates, which means moves, allowing only a certain substance or substances to cross the membrane. For example, glucose carried in blood and needed by red blood cells or cellular activities enters these cells rapidly through specific transport proteins in the plasma membrane. This glucose transporter is so selective as a carrier protein that is even rejects fructose, a structural isomers of glucose. Thus, the selective permeability of membrane depends on both the discriminating barrier of the lipid bilayer and the specific transport proteins built into the membrane. But what determines the direction of traffic across the membrane? At a given time, we will a particular substance enter or leave the cell, and what mechanisms actually drive molecules across membrane. We will address these questions next as we explore two modes of membrane traffic: passive transport and active transport. Thanks for listening.